Okay, in this video we are going to go into some detail regarding the basic workflow and analysis for genomics type studies. We've talked about these studies previously in terms of what kind of data they can give us and generally what they are, but here we'll dig a little bit deeper in terms of how this whole process actually works. Before we do that though, I want to do something called activate your prior knowledge. So in this image here on the right, it's showing roughly how long you'll remember things if they're in your short-term versus your long-term memory. My goal always when we are learning together in class is to get any information that we're discussing from that short-term to your long-term memory. One way to help facilitate that process is to activate your prior knowledge before we give you any new information. Essentially, that means any new information you're learning is going to have an anchor point in your brain and it'll be connected to what you already know, and that'll make it much more likely to stick in your brain and move to your long-term memory. So, first, what I want us to think about, because like I said, we have talked about high throughput sequencing previously in this semester, so what I want you to do first is just think about what is high throughput sequencing, or we often abbreviate it as HTS. So what is it? I'll give you some time to think about it or jot down some notes on your own. Okay, so high throughput sequencing, or again HTS, it's also sometimes called next generation sequencing or NGS. But what this refers to is non-Sanger-based DNA sequencing technologies. So Sanger is another thing we've talked about previously. That's where you're sequencing one gene at a time. So Sanger sequencing is something that gives you these chromatograms like this, where each color represents a different nucleotide, and then you're able to deduce the sequence that way. High throughput sequencing is not Sanger sequencing, but because it's not, what it can do is give you millions or billions of DNA strands sequenced at the same time in parallel. That's why it's called high throughput sequencing. And so that gives us much more throughput than Sanger technologies, which again are only doing one gene at a time. So high throughput sequencing can look at every gene in the genome simultaneously with this technology. Another activate prior knowledge question for us: what are the types of genome high throughput sequencing we've discussed previously? I believe this was way back in our molecular markers module at the beginning of the semester. What are some of the types of genome sequencing we've talked about previously? All right, so one of the ones we've talked about is called reduced representation. An example of this that we've talked about several times is RAD-seq. So reduced representation is, instead of looking at the entire genome, it's looking at still many thousands of markers like SNPs, for instance, spread across the entire genome, but you're not looking at the entire genome. Another type we talked about is targeted sequencing. So that's something like metabarcoding that we've discussed before, where you're using high throughput sequencing techniques to sequence a bunch of markers at once, but you're only looking at one particular marker, say a CO1 mitochondrial marker, for instance, across all of your different samples. So it's targeted in the sense you're only looking at one locus, but you're still able to sequence many of that loci across many samples at the same time. And then the third type we talked about is whole genome sequencing, and we often abbreviate that as WGS. And that's where you're sequencing everything, all of the DNA in the organism, the entire genome. All right, so let's talk now about the basic workflow and analysis when you're looking at high throughput sequencing genome studies. So how do you actually do that? And I will say we're going to go into some details, but not all of the details. So there's another course here at Sac State taught by Dr. Visker. I know at least one of you is in this class right now as well, where that'll give some more details about the analysis side of things. So we'll go into some, but not all details, just as a heads up. So the first thing you want to do when these types of studies is extract your DNA. So if you're working with the snails like I do, what we do is often take a piece of tissue from the foot, so you can use sterile scissors, snip off a little piece of tissue, and then use a DNA extraction kit. Oftentimes those involve a column. You do the process of this DNA extraction, and what you end up with is your liquid with your DNA sample inside. So at that point, you've extracted your DNA. Next, what you want to do is fragment the DNA into small pieces. So here we have our original whole intact sample DNA. You can then fragment that a couple different ways, either physically, if you do a bunch of mechanical shaking or shearing, that can actually fragment the DNA, or you can also do enzymatic means where you have enzymes in there that are chopping up the DNA, like we talked about with rad sequencing, where you're using restriction enzymes to chop up the DNA. So at the end of this stage, what you have is fragmented DNA, where now it's in smaller pieces. The next thing you want to do is make your DNA sequencing library. And this we've talked about a little bit before as well when we were talking about demultiplexing with the garter snake male and female data in DNA Subway. So when you're making your sequencing DNA library, one of the things you want to do is you attach your indices. And remember, we've talked about this previously. So an index is a unique nucleotide sequence that's usually like 6 base pairs long. So here we have index 1. Actually, let's do this. Here we have index 1 and index 2. So those sequences of the indices are different. So when you have your DNA that's been extracted from samples from different populations or male or female snakes, for instance, what you can do is attach those unique indices. So say we want to use index 1 for all the male snakes and index 2 for all the female snakes. So once you've attached those indices, you can mix everything up. When you sequence it, you're going to get a bunch of different sequences back that are all mixed up. We don't know which sequences came from which original sample until we do that demultiplexing step, where now we can pull out all of the samples that had index 1, all of the samples that had index 2. So that's why this step of attaching indices is so crucial for making our sequencing library. It allows us at the end, when we've done all the sequencing, to sort all of our sequences to determine which sequences came from which of our sampling groups. Another thing you want to do when you make your sequencing library is attach something called adapters. So in this image here on the left, we have our genomic DNA that's been fragmented. Remember, we talked about that. And then we have these adapters that we're attaching to those fragments of DNA. And now that's shown here at the bottom. The reason why we attach these adapters is to allow our DNA samples to actually bind to something called the flow cell. So I'll show a picture in a couple slides of the actual DNA sequencing machine. This machine contains something called a flow cell, and your DNA samples have to attach to that flow cell by binding with complementary DNA sequences. So these adapters, again, are DNA sequences just like the indices we just talked about, but they're designed specifically to be complementary to the flow cell. So by adding these adapters, we're ensuring that our samples are going to bind to that flow cell. Only things that are bound to the flow cell are going to be sequenced. So we have to do this step to make sure we're actually going to get sequence data from the samples we're preparing. The adapters ensure that the samples are going to bind to that flow cell. The next thing we want to do then is sequence the actual DNA library. So this is one example of a sequencing machine. Illumina is one of the most frequent companies that's used today. The flow cell is somewhere in here. I don't know, I've never actually used one of these sequencing machines. I usually pay people to sequence it for me because we don't have sequencing machines at Sac State, but this is what the overall machine looks like. Once you sequence your samples and get your data back, your data comes back to you in the form of these really short reads, between 50 and 250 base pairs long. So that depends on which particular type of sequencing technology you're using, but overall, all of the sequences you get back are going to be much shorter than the original full-length gene sequence. So what you have to do with that data is reassemble it into the original DNA sequence, the original gene, full-length gene sequence that it came from. So that's illustrated here. We have our full DNA molecule that we fragment into small pieces. We sequence each of those small pieces, and then what we do for this assembly step is look for overlapping DNA sequencing. So it's kind of like when you're putting together the ends of Lego blocks, for instance, you're fitting them together. And this is all done by bioinformatics programs, but what you're doing is essentially looking for these regions of overlap. Once you have these 3 short reads now that have been identified to be overlapping on the ends, you can reassemble this full-length sequence now. So that's this reassembly step. Because the reads you get back are short, just as a limitation of the sequencing technology, you have to do this assembly step. Once you've reassembled that sequence, what you want to do is annotate it, and that annotation step figures out what DNA region or function that reassembled sequence represents. That's often done using databases like GenBank through NCBI. So the overall idea here is that you have your unknown sequence. You just reassembled it, so you have the nucleotide sequence, but you don't know what gene it's associated with. You can enter your unknown sequence into this GenBank database. GenBank contains this huge repository and database of known sequences. So they have sequences where they have the nucleotide sequence, and they also know what gene it corresponds to. So you can enter your unknown sequence into that database, and what it'll do is return for you different possible matches. So here it's showing you for your unknown sequence and whatever match it found in the database, how well those 2 sequences match, and it'll tell you what that particular gene is. So this particular example, it's matching an RNA terminal phosphate cyclase. Okay, so, and then you do this through bioinformatics programs, because remember, when you're doing this type of genomic sequencing, you're getting thousands and thousands of reassembled reads. So you don't do this one by hand. You do it all through bioinformatics programs, but the overall idea is that you're comparing your unknown sequence to a database to figure out what the actual DNA region or function of that reassembled sequence is. So to summarize those last 2 steps, here's an image showing the assemble and annotate. So we have our raw reads, which remember are short, 50 to 250 base pairs. We're reassembling those, so looking for regions of overlap. And I have here assembling de novo. De novo basically means from scratch. So if you are sequencing a new organism that has not been sequenced before, there are no genomic resources for it. You have to do this process of reassembly from scratch by looking for these overlapping regions we've just talked about. So the snails that I've worked with, that we've talked about several times throughout the semester, I had to do de novo assembly because no other researcher had done high throughput sequencing with that species before. So that's what de novo means. So we're looking for those regions of overlap to reassemble our full-length sequences. So in this case, we've reassembled 3 different regions. This is just for example purposes. Remember, you're doing this for thousands of different regions. But in this example, we have 1 green region, 1 red region, and 1 blue region. And then when we do this annotation step to figure out what genes each of these different regions correspond to, we might find something like this blue region is a heat shock protein. Oops, that's the wrong color. The blue region is a heat shock protein. The green region is a stress granule gene. We'll talk more about these genes later. This is a little teaser. And then we might find for this red region, it doesn't actually correspond to a gene. It corresponds to a noncoding region like an intron. And that's because, remember, if we're doing this genomic sequencing, we're sequencing everything, both coding and non-coding regions. So oftentimes what you'll get when you're trying to do this annotation step, you won't actually find an annotation for some of your gene regions, and that's because it's in a non-coding region where we actually don't have a good understanding of what that particular region of DNA is doing. Lastly, what you want to do with this type of sequencing info, we've talked about this previously as well, is investigate nucleotide variation. So you might want to find SNPs, for instance, between Northern and Southern California at this particular locus, which is illustrated here. And when you're identifying those SNPs from your sequencing data, that allows you to look at genome-wide population structure to see if there are differences among populations. It allows you to identify outlier loci, to identify local adaptation. So you can do several different things with this nucleotide variation data that you get from your high-throughput sequencing genomic data. And lastly, in this video, I know this one's a little bit long, apologies. I want to talk about a few examples. So we've talked about what high-throughput sequencing looks like in terms of the workflow for genomics. I want to leave us with a few examples of what information you can actually learn from this type of whole genome sequencing. So this image, these two images here are illustrating two types of oyster species where they've done whole genome sequencing. And what they found is actually that there are a lot of gene duplications in these different oyster species. And this is really interesting because the particular genes that have been duplicated in these species are related to functions like environmental stress response and immune function genes. And so oysters are living in this really stressful intertidal environment. They're also sessile, which means once they're in that environment, they're stuck. They can't go anywhere. So they seem to have evolutionarily developed these really cool strategy where they've actually have multiple copies of these genes that allow them to tolerate these environmental stresses. So that's really cool. And they found this across multiple oyster species. I have the references down there. Okay, and then lastly, you can also, with whole genome sequencing, look at non-coding regions. Like I just said a few slides ago, when you're getting data from whole genome sequencing, you're looking at both coding and non-coding regions. Those non-coding regions can be really interesting when you're looking at promoter regions, for instance. So just as a quick refresher, if you have a gene here, you also have a promoter region before the gene, and you have things like transcription factors that have to bind to that promoter region to allow that gene to be expressed. In the copepod that I'm showing here, this is the main study organism for the lab I did my PhD in. I was the weird one that worked on snails. Everyone else worked on copepods, which are shown here. Plankton from SpongeBob is also a copepod, if that helps you. So in this paper, the citation is down here. What they found was that in different populations of this copepod, in Northern versus Southern California, the sequence of the promoter was different in these different geographic regions. So essentially, in some populations, the sequence of the promoter was a better match to the transcription factors, and so that meant it was easier for some populations to express high levels of heat shock proteins, for instance, to allow them to better tolerate and respond to environmental stresses. So this is super cool. We'll talk later about the gene expression itself, but this information from the promoter regions and how that matches to the transcription factors, that is only possible by looking at this whole genome sequencing approach where you're getting information about these non-coding regions. Because the promoter region is non-coding, so you're only going to get that type of information if you use a type of sequencing approach that allows you to look at some of these non-coding regions.